Hi, and welcome to Faces of Gene Therapy. I'm Erik Paulsen, chair of the Institute for Gene Therapies. Gene therapy is no longer a distant scientific promise. It is becoming a real world reality for patients and families facing some of the most serious and rare diseases. But innovation alone isn't enough. Getting these life changing therapies from the lab to the people who need them requires smart policy, thoughtful regulation, sustainable payment models, and most importantly, listening to the voices of those most affected. In this series, we'll bring together patient advocates, scientists, clinicians, policymakers, and industry leaders to explore how gene therapies are moving from discovery to delivery and what still stands in the way. We'll have candid conversations about regulatory pathways and newborn screening, access and equity, defining value, and how collaboration across sectors can accelerate progress. Each of these conversations will focus on a core challenge or opportunity in the gene therapy ecosystem, with one goal in mind: ensuring that these transformative therapies reach patients quickly, safely, and as equitably as possible. Thank you for joining us, and welcome to Faces to Gene Therapy. Our first guest today is Dr. Allyson Berent. For Allyson, science took on a whole new meaning the day her daughter was diagnosed with Angelman syndrome. Today, as Chief Science Officer at the Foundation for Angelman Syndrome Therapeutics, also known as FAST, she's turning that urgency into progress, advancing the first gene therapy programs for Angelman syndrome and reshaping how rare disease research moves from discovery to treatment. Allyson also co-founded AS2 Bio, a drug development accelerator that is building a new model for how science and philanthropy and industry can be working together to deliver treatments faster. So let's dive right in and hear more on some of her perspectives on the pressing issues in gene therapy today. Allyson, thanks for joining us. We really appreciate it.
Thanks for having me. I'm happy to be here.
Maybe we'll just start with this. Gene therapy is advancing rapidly, really fast across many rare and monogenic conditions. From your vantage point, leading FAST's scientific strategy, what really excites you the most about the potential for how these technologies are now able to fundamentally change outcomes for patients with genetic disorders?
Yeah, I mean, I think it's really there's two sides to that coin, right? It seems to be moving very fast, except it's moving at a snail's pace to those of us who are living with the disorders. And so it can never be fast enough. And I think that's really the balance of why the ecosystem is changing so much and why a lot of different types of funding streams and different types of groups are starting to take it on themselves. And that's helping to expedite the progress and the process of how to develop gene and cell therapies for rare diseases, is because what traditionally would be considered fast when you're living every day with the disorders really feels like it is just not fast enough. And so I think that's kind of where it starts. What's really exciting is the fact that there are more and more groups that are surrounded by expertise that understand the technologies, whether it's some of the more novel technologies or some of the technologies that have existed for decades that just have not reached patients in a really great way or in a grand way because of the roadblocks related to various different things, whether that's regulatory roadblocks, whether that's financial roadblocks, if it's manufacturing roadblocks, or just interest by the field. And as more and more people are starting to jump in with just single disorders and say, you know what, if no one's going to do this, I'm going to do it, then suddenly the competition gets vast, the opportunity is greater, and really, at the end of the day, patients can get treated. And that's what we all want. And so what really excites me is the visibility to gene and cell therapy. What excites me is the miraculous opportunity that scientific marvels can actually change the trajectory of a disease that was considered completely debilitating or fatal, and maybe no longer. And because so many of these therapies have been in exploration in human clinical trials for the last decade, they're all kind of coming to the top right now. It is really in the last 6 months to the next 2 to 3 years, I think there are going to be more gene therapy approvals, hopefully, than any other time. And part of that is not necessarily the changing ecosystem or the changing administration or regulatory framework. I think it's because there have been hundreds, if not thousands, of gene therapies and clinical trials, and they all started around the same time, and they're all coming to a close around the same time. And as we see all of these cell therapies, like the hematopoietic stem cell ex vivo autologous gene therapies, that we've all been talking about for years, that they're truly curative. And none of them have really hit the stage until the last 2 weeks. There have been 2 gene therapies either approved or have gotten incredible media visibility to show the potential for cure for disorders that nobody would think had a chance, like, mucopolysaccharidosis or cystinosis, or, some of these central nervous system disorders that no one really understood how a bone marrow transplant could benefit them in a really important way. And so all of these other programs that are following their trail, hopefully, are going to have a real path to understand how to get through approval, how to get through the toxicology programs to get to human, how to get through the human trials in a way that's affordable, understanding the safety that's required to get there and to get to patients. And I think because of the incredible work that's been done before most of these newly diagnosed disorders, the path for these disorders that are coming online is going to be much faster, much more efficient, much less expensive, and much safer, I hope. And that will really transform the field. So what excites me the most is that we can finally leverage the incredible work that has been done before us in order to take that and try to create the roadmap that we're not all just reinventing the wheel and living in a silo, but we can actually share, borrow, and steal from each other and really advance the field in a meaningful way. And I feel like now is the time that's really happening, and that really gets me excited for the field of genetic disorders in general, not just Angelman syndrome.
It's pretty clear with the excitement in your voice that there's a huge promise for patients coming ahead. There's no doubt.
No doubt, no doubt. We have to just hope we could afford it. We have to hope we can raise the money to do it. And we have to hope that the regulatory landscape allows for this progress to be made and can leverage the learnings of others in order to not make everyone have to learn it again.
So the Angelman community has really helped pioneer a new model for accelerating gene therapy development. How do some of the lessons that you've learned about the urgency and the coordination and the de-risking on early science translate now into other rare disease communities facing similar unmet need?
Yeah, part of our mission, of course, is to bring transformative, very meaningful treatments to all of those living with Angelman. And Angelman syndrome is a really unique disorder. It's incredibly elegant genetically, while incredibly devastating genetically, and phenotypically. And so because it's commonly rare, so it affects approximately 1 in 10 to 15,000 people. That could be 500,000 to a million people in the world. So it's rare, but it's not considered ultra rare. And because of that, there's been interest in Angelman syndrome. And because of the genetic elegance, you can unsilence a silent copy of the gene, or you can replace the gene that's missing. And because of that, it makes every genetic technology applicable to Angelman syndrome. And so when you are an organization that leads by mission, which is to come up with the best possible way or combination of ways to truly transform a single disorder, what you can do is you can invest in all of the ways. And by doing that, by knocking down a silencing mechanism, by replacing a protein or a gene, by doing that through an ex vivo autologous gene therapy, through an in vivo AAV gene therapy, through a CRISPR gene editor, through a CRISPR activator, through a zinc finger, every technology that's ever touched genetic disorders applies. So because of that, and because we have the incredible strength of our community to financially support it, we are able to throw everything at this disorder and see what sticks. Or what combination of things that stick, right? Because likely there's not one size fits all. And so because of that, it allows us to serve as a model for every technology, for every genetic disorder. And if you can use one disease that has a clear path for a clinical trial, that has a clear regulatory path for potential approval, because we really deeply understand what's meaningful and measurable in this disease, and we have clinical trial experience with a 20-year natural history study, longitudinal, to be able to compare that as a comparator, you are able to then test head to head, essentially, different technologies. And now you can use that as a model for other monogenic disorders to follow in that path, to say, okay, there's a regulatory path for this disease with a CRISPR editor. There's a regulatory path with an ASO. There's a regulatory path with an AAV gene therapy. There's a regulatory path with an HSC gene therapy. And so what is best for that one disease? Most disorders have one option. Well, we have about 30 options. So it allows each disorder to take a similar path and borrow what we've learned and share that with others so that they're not spending 10 years or 12 years or 15 years, but they can do it in a more expedited way and really avoid some of the landmines that we hit, because we were one of the first disorders to actually do this in such a big way, not only to fund an academic lab for proof of concept, but actually to fund all the way through to a phase 1/2 clinical trial in which we can partner that for a phase 1/2 trial and partner that for a pivotal trial for a potential approval. And so that allows us to really understand, where does the mouse fail us? Where does it predict what's going to happen in human trials? What do we need from a toxicology perspective to feel comfortable? What do we need to, how many neurons do we need to hit in order to really have a profound clinical benefit, not just a benefit in a mouse model? And so because we are committed not only to help those living with Angelman syndrome, but we are very committed to share our learnings with every other disease that can benefit from what we have learned, that allows us to de-silo drug development for rare genetic disorders that can expedite their process and avoid the landmines we already hit and ensure that they have success too. Because really, when you only have one or two options, the chance of success is low. But the reason it's low is not necessarily because science failed. The reason it's low in gene therapy or genetic therapies is because people run out of money. Priorities change. And if we can save people time and money, then maybe they would actually see the light of what is possible for their disease. And if that means that Angelman was allowing them to create a faster path, then we've done our job. And the goal in life is impact, right? And if we can have an impact on Angelman syndrome, that's my dream. Of course, I have a daughter that lives with it. But if we can have an impact on 7,000 other rare genetic disorders, or at least 1,000 other neurodevelopmental disorders, then what we have done has really just completely expedited the path for all. And that's really what we're about, is to try to make sure we don't keep what we learn to ourselves and we share it.
We've already kind of touched on this, but I do want to make sure I ask a question here. As multiple platforms emerge, from gene replacement and gene editing to RNA-based approaches, how do you see the field moving towards providing that toolbox, a toolbox that can reach diverse patient populations and ages and disease severities across the rare disease spectrum?
Yeah, we have to. We have no choice. We have to provide a toolbox because many people nowadays are coming at this with zero scientific knowledge. They're coming at this as a parent or a loved one who doesn't have a pharmaceutical company, a researcher, or a team with millions of dollars, if not billions, backing them. So we have to create a path in which there's advisors, in which there's expertise, whether that's regulatory, whether that's toxicology, pharmacology, manufacturing. There has to be a cookbook that has the ability to be nimble, but also the ability to be able to follow so that everybody that wants to can be successful. And for me, that's platforming. You can create platforms in which companies, rather than just working on the platform of one technology, can work on a platform of like diseases. And that's what our accelerator is doing. We're working on one disease, and the platform is Angelman syndrome. We are modality agnostic. We are disease specific. So the platform we have is understanding the models, understanding what we need to measure, understanding how to get there in the fastest possible way by borrowing from ourselves the information. If we could do that with multiple disorders like Angelman syndrome, and other people can do that with multiple disorders like the other ones they were successful in, then I feel like the whole field will advance much more rapidly. So I think you do need to create these toolboxes to understand, okay, so we're working on a CRISPR program. What is the right way to do bioanalytical assessment of all of the components for an in vivo CRISPR editor? Right? Most CRISPR gene editing, if we're really going to impact thousands of diseases, are going to be in vivo, not ex vivo. in vivo CRISPR gene editing is really in its infancy. What do we do to share and learn from one another? How do we measure the guide? In what tissues? In what fluids? How do we understand biodistribution? How do we understand how long we need to test animal models for? How long the toxicology programs need to be? How do we share that information so every disease can pile on and borrow from each other those learnings? And if one disease, took 6 months, another disease should take 4 months, another disease should take 2 months. And let's get down to the nitty-gritty. What do we need? What do we need to get there? What is enough information that provides enough safety to be able to do the true experiment, which is the human experiment? And we get real stuck on, well, this is the way it was done 20 years ago. This is the way we have to keep doing it. And I think that that's what we're realizing in genetic therapies is that's not true. We really need to grow with the field, and we need to morph into efficiencies in order to help more diseases, because the time it takes costs money and we lose lives. And so we really need to be efficient in how we do it. And we need to bring everyone to the table and create that toolbox in order to understand for each modality what's critical, what's a nice to have, what's a must have, and then get buy-in from the regulatory authorities on those studies, because that's where most things fail, is in those early discovery periods for these rare diseases. And then when we get into a clinical trial, then that's the next set of failures, right? Is who can afford to dose 150 patients with a gene therapy? Well, an ASO needs 150 patients, right? And ASOs are far cheaper to manufacture, but a gene therapy can't afford to do that. So what do we need in order to understand a path of success? And how do other diseases borrow from that information rather than have to go and have the same conversation again and get different feedback from different people in order to understand how to best design these trials? And then we all have to speak to the payers, right? How do we ensure that SMA is paid for, but Angelman's not paid for? Why? Well, they're, on a newborn screen, and the difference in survival is so profound, where in Angelman syndrome, maybe the difference is in self-feeding, or maybe the difference is in activities of daily living, because it's not a deadly disease. So therefore, the thresholds are different on what we would consider a success. But who's to define the success, right? So we all have to learn about these different disorders. If it's cancer and you're not living, but you're living a few days longer, is that enough? You're living a few months longer, is that enough? A few years longer, is that enough? Well, what happens with neurodevelopmental disorders? What happens with non-deadly diseases? But there's no other alternative. What's enough? We need to learn from each other and try to define that in a way that really makes sense. And I think when everyone goes at it on their own, independently, I think that we slow things down and we waste a lot of money.
Just as you laid out, many genetic conditions share the same barriers, that slow translation, limited funding, and fragmented infrastructure. How can the progress made in Angelman syndrome serve as a proof of concept for accelerating timelines and improving access for thousands of other rare diseases?
Yeah, I think it can, and I think it will. We're still early. We have our ASO that we developed out of the foundation that we supported through a company called Genetics Biotherapeutics into a phase 1/2 that was acquired by Ultragenyx Pharmaceuticals prior to phase 3. And I think the fact that we did that as an organization from a foundation starting point allowed us to be able to share so many of our learnings in ASOs for non-deadly disorders, right? Before us, very few ASOs were approved for neurologic conditions. And I don't think we can use Spinraza and spinal muscular atrophy as our example, because really that's not a brain disorder. So really, Angelman was one of the first non-degenerative brain disorders that really took it on. And what we learned, and with 2 other ASOs following behind, with a program with Ionis and then a program from Roche and now Oak Hill, what we've learned is that you can change the trajectory of neurodevelopmental disorders. Look at this. There are kids that were not self-feeding that are self-feeding, that were not potty trained that are potty trained, that were nonverbal that are speaking words or using signs or using an iPad to communicate better, that can follow instructions and not walk into a street and get hit by a car. I mean, these are life-changing things that are happening to humans well after birth. Those learnings are now allowing us to realize in the field of neurodevelopmental disorders that we need to come up with a way to granularly measure change. And so all of the things we do from a clinical trial perspective are through our pre-competitive consortium called the Angelman Syndrome Biomarker and Outcome Measure Consortium that myself and my colleague Jennifer Panagoulias lead out of FAST. And we share everything we learn and everything we fund. I mean, we put $5.1 million into developing endpoints and biomarkers for Angelman syndrome, which is just one example of a neurodevelopmental disorder that many are misdiagnosed as because they're so similar. So everything we have funded, we share. We share our clinical trial designs. We share all of our endpoints. Any endpoint that we've made from scratch or we've modified that FAST has funded, we give freely to anybody that wants to use it. Because our feeling is that nobody should redevelop what we have already spent money on. That doesn't make sense. And then our learnings from that, which we have beautiful engagement with the FDA on, we share. So we share the feedback we get. We have a meeting every year. We invite all of the other neurodevelopmental disorder rare disease groups to, so they can hear from the regulatory authorities. They can hear from sponsors. They can hear from clinicians how these measures are behaving in clinical trials, how we're working through biomarkers, through CSF biomarkers and blood biomarkers for a brain disease in which it's not deadly. So we don't have degeneration. We don't have proteins like NFL in CSF fluid that we can measure like you do for some of these other disorders because it's not degenerative. So we have to be creative and thoughtful in other ways we can measure. And so we do that through EEG or through different protein signatures or through different measurements of the deficient proteins. And we share it. If we fund it, we share it. And that's kind of our model. And then I advise so many other neurodevelopmental disorders on how do you best design your toxicology study, your pharmacology study? How do we marry your proof of concept study to be a minimum effective dose study, to marry that to a toxicology study in a way in which it doesn't have to be 2 years? Can we do this in 6 months? Can we do this in 9 months? How do we ask the right questions in order to be able to get the answers that we can do this in an expedited manner? And I think the field from 5 years ago to today has shifted tremendously. And I think part of that is due to the sharings of the Angelman syndrome, because we are always willing to share with other groups and other communities what we feel like we did too much of or not enough of, or what we could have done more of, what we could have done less of. And that helps to really kind of shave off the fat, the excessive fat, and really get down to the meat of what do you need in a package? What do you need in a clinical trial? How many sites do you really need? Who are the good PIs that can run that trial for you? Who knows how to get access with an ICM delivery to the back of the skull? Not everyone knows how to do that for a gene therapy. Should you do ICV? Should you do ICM? What are the trade-offs? And because we've learned that, we share that. And I do think it has advanced the field, or at least I hope. I'd like to believe that these efforts have advanced the field, and we will definitely continue to do that at every turn. Because we're only in our infancy. We're learning more day by day. When our HSC program gets into the clinic, we're going to learn a lot. When our non-viral delivered CRISPR gets into the clinic, God willing, we're certainly going to learn a lot. We're certainly going to learn a lot, and we're going to share every step of that.
You referenced the pipeline of new therapies that are in trial right now, going through the FDA process. With momentum growing in Washington and across the industry, what do you believe policymakers and developers need to prioritize right now to ensure that gene therapy fulfills its promise, not just for Angelman syndrome, but for those millions of patients that are living with rare genetic conditions who lack meaningful treatment options today?
Yeah, for me, there's so many pitfalls, I think, in where people get stuck. And I think a lot of that is policy and regulatory. Certainly, it's not all of it, but it's at every stage. It's at the preclinical stage, it's at the clinical stage, and it's at the approval stage. For me, what's the most important thing, and the thing that I see the most, is that people that don't live with a disease, regardless of what it is, really don't have an unbiased ability to judge what's meaningful, to judge what should be really considered change. They certainly have an ability to judge what's measurable. Statistics are statistics, right? Anybody can look at statistics in a creative way and tell us how to measure something in a way that makes sense. But what they can't tell us is what is meaningful. And I think what people get stuck on is very easy in cancer to say tumor burden is less, survival is more. I feel better because I can speak and tell you I feel better. But when you have these children, and most rare diseases are neurologic rare diseases of nonverbal children who are living in a debilitated manner, and their proxy is their parent. What nobody can tell me is what matters to me, my child, her sisters, and our family. And what I can tell you is that people need to listen. They need to listen to what truly matters and affects our lives, because if they really listen and they really care, they're going to realize that what could be a small change in their neurotypical world with their healthy children is an enormous change to a child who can't feed themselves, who can't use their hands, who can't point to something to say they want this or that, or this or that hurts. And suddenly you have a kid who has a finger point, who can now use an iPad to communicate and tell you what matters to them, what hurts them, what they need, and what they want. For a kid who doesn't have a single word, who can say one word, and that word's help. Because when their parents are no longer around, they can scream for help. God forbid if they're being abused, our biggest fear in life. Those are the things that matter. But what the general public, and I think policymakers, understand in their own general neurotypical life is that it's life or it's death. It's black or it's white. It's survival or it's lack of survival. But there is a gray for most of us that only we can speak to. And if you don't listen, you're not going to understand what meaningful change truly means. It's my kid having more independence. It's my kid having semblance of a happy life, that they're not reliant on an adult for absolutely everything. But that doesn't mean they either are reliant on an adult for everything or they're reliant on an adult for nothing and they're completely independent, because there is a spectrum in between that is so unbelievably meaningful to us parents and family members that people need to listen. And I do believe that policyholders are starting to realize that. But I think when they hold rare disease to statistical significance of a p-value of 0.05, and what they need to understand in these global diseases is that, you know what, in Angelman syndrome, 85% of kids don't sleep. My kid sleeps. She's in the 15%. So if sleep improves the lives of the gene therapy and independence and communication and cognition, but my kid already sleeps, she has no change. How do you measure that in a global disorder? She doesn't have a lack of treatment effect. She already was sleeping normally. So how do you measure that, right? People try to do these composites, and they need to understand every kid is different. Every family is going to have different expectations, and every family has different dreams. And if we can measure meaningful change to that one kid, which shouldn't necessarily be in means and medians and statistics, because my kid is going to look different to this other child who sleeps 3 hours a night, and a meaningful change to that kid is sleeping 5 hours a night. Well, my kid sleeps 11 hours a night. Don't hate me, but she does. So therefore, sleep is not on my list of most important things, but independence is. And I want her to be able to have more independence from adults so that in life, when I'm not here, she can do more for herself and not need full-time adult independence. That's what I want for her. But a kid that doesn't sleep more than 3 hours a night from age 0 to age 30, guess what that parent wants? They just want a full night's sleep. That's all they want. So the policyholders need to think about this in rare disease. It is not one size fits all. It is generally not life and death. It's debilitating diseases that want an improvement in life. How do you measure that? And if we spend the next 2 decades trying to figure out how to measure it, we're not going to make any progress for these diseases, right? We just have to open our minds and open our ears and listen to those impacted, and then make judgment based on the voice of the patient, not necessarily the voice of a biostatistician. And I think that's where in policy we get a little stuck. And I don't think people want to get stuck, but they want to be sure they're making the right decision, which is usually erring on conservatism, not erring on the likelihood of success, right? It's the likelihood of failure. And I think that's where us as rare disease families who are really working hard to show who our kids are and what their potential change is feel that we get stuck.
Well, and thank you for sharing all your thoughts on that value component, which we talk a lot about at IGT. Allyson, we really appreciate you sharing your insight, your thoughtfulness, your expertise in this field, and thank you very much.
Yeah, it's my pleasure. I'm always happy to help. And anything we can do to advance the field outside of Angelman syndrome, of course, we're here to do it because it took us too long, which all of you think is fast, right? We got into clinical trials quickly compared to others, but it never feels fast enough. And if we can make sure that others can move more quickly and more safely, that's really what we're here to do.